Hello, my name is Gretchen Reifenberger, and today I will be talking about the development of methodology for DNA extraction and pathogen detection from fed ticks. Ticks are well known for Lyme disease and for their prevalence in wooded areas and also when you're camping and just watching out for ticks. And they are known to carry pathogens such as Borrelia burgdorferi, which is the disease-causing agent for Lyme disease. There's also Rocky Mountain spotted fever, which is another common disease, tick-borne pathogen, and tick-borne illness. Ticks exist all over the world. However, around 60 of them are only harmful and transmit pathogens to humans. And roughly 90 of them live in the United States, continental United States. Within the Southeast, there's about 6 hard-bodied ticks that we focus on in our lab. And with all the species of ticks, that includes hard-bodied and soft-bodied ticks. However, our lab only focuses on hard-bodied ticks. So the tick life cycle starts at the egg stage where a female lays eggs, the eggs hatch, they go to a larva. Larva then feeds on a host, and the host is typically, the first host is typically the smallest. You're typically thinking about mice or a bird or something smaller. And then they have one blood meal here. They will fall off, drop to the ground, and eventually over time molt into a nymph, and then a nymph will go and will feed again. We'll have another blood meal number 2 on a different host, slightly larger. We're thinking rabbits, maybe some chipmunks, something like that. And then it will detach and molt into an adult, which will typically feed on humans or larger mammals such as deer or dogs, and that is where they meet other ticks and where they will actually have to start the mating process, and that will allow the process to start all over again. Engorged ticks are ticks that have fed. They have ticks that have acquired a blood meal, that have fed off of their host. And this picture just shows just how large ticks are, how large they can become. Over here we have a small adult tick, and over here we have a fully engorged tick. This tick is obviously huge. It has a ton of blood inside of it, a ton of its blood meal, and because of how large engorged ticks are, it has been shown, research has shown, that it is much harder to amplify DNA, to extract DNA, and to amplify DNA just because of how much is inside the tick. It is not just the tick DNA you're also getting host DNA, and if you're trying to do pathogen detection, it is quite difficult. And because of this, there isn't a lot of research that fully focuses on engorged ticks and engorged tick DNA, and the methodology, the best way to get the total tick DNA out of the host. And because there isn't a ton of research, we have decided that we wanted to try to develop a methodology for DNA extraction and PCR amplification of total DNA from engorged ticks. And we wanted to do this by using different extraction methods and trying to figure out which is the best and which has the highest yield in DNA concentration, but also pure DNA. Our first objective is to create a set of standards to classify engorgement of ticks, and this is going to be based on length, height, and weight, but we focused the most on weight because it really depends on how much the tick has eaten, whether the tick was able to fully finish engorgement, or whether it fell off too early, or whatever the case may be. Secondly, we wanted to find the best means to extract DNA. And we wanted to find this through different methods, through different protocols. And then the last objective was to determine the best protocol to amplify DNA from engorged ticks. And this includes use of different primers, different reagents, and different tags. So here we can see a bunch of engorged ticks. Up here, we see a pretty small engorged tick. That's a tick that hasn't had a super large blood meal. Then down here, we see a most likely fully engorged tick. That tick has had all that it can eat, and it can no longer eat anymore, and will molt or will feed again. And then over here we have a slightly smaller one, and you can see just the different array of ticks, different size of ticks, and how necessary it is for us to create classifications to know if the ticks, the tick weight, tick size, the amount of blood in the tick is going to be a determining factor in different pathogen detection and amplification. However, at this time, we split apart the groups, and we are looking at all tick weight classes pooled together. So we weighed all of the ticks. We weighed around 200 ticks and got their size and their height, and we have looked at 72 of them right now. So majority of them were pretty low on the weight, pretty low weight rise, and we had just a couple that were higher weight and towards our larger ticks. In our second objective, we wanted to find the best way to extract the DNA, and because of this, and due to this and different preparation methods. So our first preparation method is quartering. Quartering is pretty common. It's down here, a picture of you cut a tick into 4 quarters, and that's pretty much, you go from there. With internal organs, we wanted to try to do this in order to kind of get away from that hard-body tick. Since we do work with hard-body ticks, their external surfaces can be pretty hard to penetrate, so we decided to go for the internal organs. The internal organs are including but not limited to the rectal sac, the midgut, salivary glands, the trachea, and the ovaries in female ticks. And then our first extraction method was cetyl trimethyl ammonium peroxide, or CTAB, and that was a method that we'd had found in research that had been used with engorged ticks before, and we decided that it would be an excellent opportunity to continue to look at how it works with engorged ticks, if it does work productively. And then with phenylchloroform extraction and the commercially available QIAGEN Easy Blood and Tissue Kit, both of those are proven over time and throughout lots of research that they work really well with adult ticks, adult non-engorged ticks. So we decided to see how they would work and how they would function with engorged ticks. So this is the DNA concentration of all of our sample ticks, and there was a very vast difference of data we were able to have. So with CTAB internal organs and QIAGEN internal organs, we're seeing lower numbers of DNA concentration, while phenylchloroform over here is 1400, its concentrate of 1400, which is pretty incredible. So overall, we were pretty happy with how much DNA came out of them and how much DNA we were able to extract. Excuse me. And then we were also pretty happy with how pure our DNA was. Okay, so around 1.8-ish is considered pure in DNA, and that is nanograms per microliter. And those are pretty pure. Around 1.6, we're getting into some more unpure DNA, and anything below that is pretty impure. And then around over 2, is probably due to protein in the DNA and just lots of protein in the DNA. And under 1.6 is probably alcohol contamination. So we see that that could be the case down here with phenylchloroform, internal organs, or with the QIAGEN internal organs. But overall, pretty satisfied with how pure our DNA was. Our third objective was to see the amplification of our samples and see how the different reagents we could use and to find the best protocol. And through this, we were going to test different reagents and different Taq polymerases to see which ones worked best with which Taqs and how we could amplify as productively. But because of coronavirus and because of COVID-19, we were unable to access our labs, and we were not able to test our primers or our Taqs. As you can see in this gel down here, we actually see some amplification here and here, but we wanted to try different types of Taqs, and this is just Go Taq. We wanted to try HemoClean and AmpliGold. Overall, we had strong amplification. Our amplification was strong with QIAGEN, as of 75% of the time, QIAGEN was amplifying on our gels. And then we had the lowest with phenylchloroform. Around 25% of the time they were amplifying. And again, this is with all of our tick weight classes and everything pooled together. Overall, phenylchloroform had the highest extraction of had the highest yield of DNA concentrations. We saw that on the graph earlier. It was very clear that it was a lot of DNA and lots of concentration. With CTAB, we found that overall they were the purest. They were averaging around 1.849 for quartered and 1.8 for internal organs. And then the QIAGEN D&EZ kit had the best amplification. We saw that just now with 75% of the time amplifying. That's very strong. And then also quartered chicks had higher amplification than internal organs. And that leads us to the fact that quartered chicks that were extracted by QIAGEN have the best amplification and higher levels of DNA concentration. Our future work, we want to test out different primers and different tacks. As I mentioned before, we want to test out HemoClean, AmpliGold. We want to test out those tacks and see how they react and if we have more amplification or we have less amplification. We also want to analyze our amplification and extraction based on different weight classes. So in all of our results, all of the different weight classes were pooled together, and we want to see how weight is affecting the amplification and extraction. And then additionally, we want to continue to run more gels for unengorged and engorged ticks and kind of look at the differences between unengorged and engorged ticks when it comes to DNA concentration and amplification. Lastly, I would like to thank Dr. DeLacy Rhodes for her mentorship and for allowing me to work in her lab, the Synovus Undergraduate Research Grant for funding this research, Morgan Torres and Fort Campbell Fish and Wildlife for allowing us to sending us tick samples and deer tissue and all of their support. And at this time, I will take any questions at this email. Thank you very much.